Good afternoon. My name is Christina Zielinski, and I would like to welcome you all to today's ESDR Sweet and Sour Kitchen Seminar. So my co-chair, Knut, Schäkel, and me are very delighted to have two outstanding speakers today, Clare Bennett from London and Dan Kaplan from Pittsburgh. Both are very famous for their work on skin immune regulation, with a particular focus on Langerhans cells, maybe the most controversial and versatile cells that we know as skin scientists. And we are very excited about what they have to teach us today about this very interesting cells in settings of immune regulation as well as immune dysregulation. So we will start the seminar today with Clare. And it's my honor to introduce her. She's a professor of cellular immunology at the Department of Hematology in UCL London. And she has started her career being a PhD student in Edinburgh, and then has transitioned to a postdoctoral position in Amsterdam with Martin Kapsenberg and Björn Clausen before becoming an independent group leader in London with various high-profile grants and funding schemes before eventually becoming a professor in London. And Clare is very well known for her work on Langerhans cells and also her famous conditional Langerhans cell depletion mouse model. She discovered, for example, that Langerhans cells can be replenished from circulating monocytes in settings of inflammation, and has greatly contributed to a better understanding of their role in settings of atopic dermatitis, graft versus host disease, and more recently also melanoma. So we're very happy to have you here, Clare. We will have a talk now of around 10 minutes, which will be followed by a presentation from Dan, who will be introduced by Knut. And at the very end, we will have an interacting discussion. So please bear with us and keep all your questions. They will be discussed and addressed at the end of these two presentations. So the stage is yours now, Clare. Thanks for coming.
Thank you very much, and thank you for the nice introduction. Okay, so yes, it's a pleasure to be here today, and I'm going to tell you all a little bit about our Langerhans cells. So Langerhans cells, as most of you I'm sure are aware, excuse me, are a unique population of immune cells. They sit in the epidermis of the skin, and they cover our body as this network of cells throughout the body. And they're incredibly highly conserved. We find them in zebrafish and we find them in mammals. And we also increasingly are becoming aware that they're important in both physiological responses in the skin, so in terms of wound healing and also interactions with nerves, and also regulation of immunity. But what exactly are Langerhans cells? So back in the 19th century, when Paul Langerhans observed these cells originally in the skin, he thought they could be neurons because of the dendritic processes. But he then revised that. He thought maybe he was wrong, maybe they were immune cells. But it was really like about 100 years later, Ralph Steinman, that has dominated the field by using Langerhans cells as a proteotypic dendritic cell. And this is because we know that these cells can be activated to migrate out of the skin. They can go in the lymphatics to the lymph nodes, and they can prime T cell responses. But even at the time that Ralph was doing this work, people like Simon Gordon and David Hume were staining the skin with their quintessential macrophage antibody F480, and the Langerhans cells were coming up positive. And this conundrum was resolved to a certain extent more than a decade ago now, when the use of mouse-like fate mapping studies in mice really showed that those macrophage precursors that give rise to all the tissue-resident macrophages throughout the body are also giving rise to Langerhans cells. So we now have a consensus where we know that these macrophage precursors are entering the embryonic skin during development. We know that around birth, it's slightly different between mice and humans, that these cells undergo a proliferative step, they fill the skin. And we know that in the adult, these embryo-derived macrophages persist throughout life. And as far as we're aware at the moment, they persist indefinitely in healthy adult skin. So we now know that these cells, which are born as macrophage precursors and are born as macrophages, somehow during this process actually end up looking more like the cells that Ralph Steinman described, more like these dendritic cells. So we've been interested for a long time in trying to understand how that is. And really important information came from our work that we did with Marta Polak, who was in Southampton. And she prepared skin biopsies from newborn babies, from infants that were more than a year old and older children. And we isolated the Langerhans cells. And you can see when you look at the frequency of the Langerhans cells, you have this dramatic increase in the cells between newborn babies and children that are a year old. But this isn't just about proliferation. When we looked at the transcriptomes of these cells, we found marked differences in their transcriptional programming. So this figure has clustered, it's calculated the predictive processes that might be occurring in the Langerhans cells at the different point based on their gene signature, and it's clustered them in functional groups. And what I want you to focus on is this cluster 5. So this is markedly upregulated in that switch from birth to the infants. And this, if we look at the genes involved, it's all about regulation of adaptive immune responses, regulation of effector responses. So you can see that these cells are becoming more like the cells we think of as dendritic cell-like Langerhans cells. And actually, when we look at the transcription factors that change during this transition, we see that at birth, these Langerhans cells express ZEB2. This is associated with specification of tissue-resident macrophages across the body, and MAFB, a very much myeloid focused transcription factor. But then after this transition, they now upregulate the aryl hydrocarbon receptor, RUNX3, the lineage-defining transcription factors that we associate with Langerhans cells. So I would like to suggest that we're beginning to have this model where before birth, perhaps these Langerhans cells, they're developing as macrophage-like cells in the epidermis, and maybe they're important here in terms of regulating skin physiology, maybe establishing neuronal networks and blood vessels. But actually, after we're born and our skin becomes an important immune barrier, we need this function to change. And now they undergo this transition to become dendritic cell-like, and they become cells that are really good at priming T-cell responses to infection. But then this led to the question for us, if this Langerhans cell identity and this switch to a dendritic cell-like identity requires this adaptation after birth, how can this network be repaired if it's damaged in adult skin? And this is an important question because in patients with blood cancers that receive hematopoietic stem cell transplants, the allogeneic T cells in those transplants will, of course, attack the tumor cells, but they will also enter healthy tissues, including the skin, and we know that they will attack Langerhans cells. And many years ago, we showed initially that the Langerhans cells actually license those T cells to become pathogenic in the skin. So using a murine model of graft versus host disease, we showed that if we delete Langerhans cells from these mice, that we no longer see this pathology in the skin. And that's also shown with the histology score, showing that the Langerhans cells are needed to activate this pathogenic effector function by these cells. But this is a still image from some live imaging that we did in our transplant model in the mice. So in this, the blue marks the dermis, and on top of the dermis, you see the Langerhans cells in green and our allogeneic T cells in red. And you see they form these really tight interactions. So those interactions lead to enhanced T cell activity, but those T cells then turn around and actually kill the Langerhans cells that have activated them. And this results in a new Langerhans cell network where the skin is now populated by replacement Langerhans cells. And this is commonly seen in patients that have received stem cell transplants. So we've spent quite a lot of time figuring out how this happens using our mouse models. We've shown that monocytes enter the epidermis after transplant, and they undergo this very programmed transition where they first upregulate EpCAM, which is a cell adhesion molecule in the epidermis, and then they acquire Langerin, which is the defining protein in Langerhans cells. And work that we did at the time with Andy Yates, who's a mathematician, where we modeled this process and we tried to understand the dynamics of how this happens, demonstrated that these EpCAM positive monocytes are very transient. They don't survive for very long in the epidermis, and by far the majority of them will die. And only 4% of those cells will go on to become the very long-lived donor replacement Langerhans cells. And at the time, we discussed how this was like when you make a Christmas card and you put the bits of glue on the card and then you cover it with glitter. And then when you shake the excess glitter off, only certain bits stick. And that led us to question, what is it about this process that allows those few monocytes to stick and become Langerhans cells? So to answer this, we went back to our transplant model, and now we sorted all of the myeloid cells from the epidermis, and we sequenced the single cells. And we could clearly see here this progression from the monocytes that enter the epidermis to becoming the resident monocyte-derived Langerhans cells. But again, what we were struck by was the heterogeneity that we find in the epidermis, where a lot of these cells are clearly not finding the right signals and actually becoming more macrophage-like cells that don't survive in the skin. So we hypothesized that maybe competition for a niche within the epidermis is the reason why we see this bottleneck and only a few cells succeed in becoming Langerhans cells. And indeed, if we look at skin sections from our transplanted mice, you can see that around the top of the hair follicles, we see this accumulation of CD11b monocytes. And this is at the point where we have this high level of EpCAM staining. So could it be that these cells that express EpCAM are sticking to these sites and receiving the niche signals? And I don't have time to go into the data today, but we used transcriptomics analysis of the keratinocytes to infer a role for jagged and notch signaling at this point. And indeed, we were able to show that if we take monocytes and we provide jagged signaling in vitro, we can generate Langerhans cells that really express a whole program of genes that we associate with Langerhans cells, including the AHR receptor and CD207 or Langerin. And we were also able to show that this signaling is required in vivo, because when we block it, we didn't get full differentiation. So what I'd like to leave you with is an idea that we have, like in the developing embryonic skin, we have these macrophage precursors that can come in and become Langerhans cells. But after birth, they undergo this postnatal maturation step that enables them to become these dendritic cell-like cells that we all know. When in the adult skin, when the network's damaged, we find that the monocytes can use this hair follicle niche to enter the skin. And of course, these hair follicles would not be present in the embryonic skin, but we have this convergent adaptation that allows these cells to find the signals in the niche and find the jagged signaling, which allows them to go through that maturation step and become dendritic cell-like cells. And really what I would like to leave you with is the thought that I think, and I don't know if Dan will agree with me, that we don't really fully understand what these Langerhans cells are doing in the skin. But I would suggest that the fact that the niche is repaired so efficiently, even when we have a man-made disease like graft versus host disease, suggests that it's fundamentally important that we have them there. And then I just finish with acknowledging my team, past and present, and then our collaborators, and of course, the people who fund our work. Thank you very much.
Yeah, thank you, Claire, for this very nice and very clear talk. In the interest of time, I'm passing on immediately to Knut, so please keep all your questions for a little bit later. They will be discussed. Knut,
yes.
Please.
Hello, everyone, dear colleagues, and nice hello from my side. We are here in the fortunate situation that we have another Langerhans cell specialist, Daniel Kaplan. It's my pleasure and honor to introduce him shortly here. He's a professor of dermatology at the Department of Dermatology and Immunology at University of Pittsburgh. He went to medical school in St. Louis, Washington University, where he studied tumor immune surveillance, and for his residency and also for the postdoc, he went to Yale University, where he then studied Langerhans cells, and I think it was in 2005 his his important publication in immunity, where he could show with different mouse lines deficient of Langerhans cells that Langerhans cells not only have an immune stimulatory role, as we saw here with Clare Bennett, that they also have an immunosuppressive role. And I think this is intriguing to have these two sides here today. Now, when he moved in 2015 to Pittsburgh, he started then a lot on T-cell immunology, and that greatly supported our current understanding of T-cell immunology in the skin. And he became very interesting in sensory neurons, in pruritus, stimulating mast cells, stimulating immune responses. And I very much like his concept of anticipated Th17 immune responses induced by sensory neurons to combat Candida. Daniel, the stage is yours. We're very happy to have you here today.
Knut, thank you for the introduction, and thank you also, Christina, for the invitation. Just making sure you can see my slides okay. Hopefully, okay.
Yes.
All right. So I'm in the unenviable position of following Clare after her superb talk, which is a very high bar to clear. And so I want to sort of start by just talking about overall what the lab is interested in. As Knut mentioned, a long time ago, we made mice that didn't have Langerhans cells. And we actually were part of the trio. I was a postdoc. We made a Langerhans cell deficient mice. Clare made a very similar mice in Bjorn Clausen's lab, and Adrian Kissen-Bennett made another very similar mice in Bernard Mallison's lab. And we sort of like had a bit of conflict because all three of us got different answers. But I'm not going to get into that too much right here. I just want to highlight that we've worked on Langerhans cells and then branched out into other dendritic cells that are in the dermis. We've also spent time looking at resident memory T cells, CD4 T cells in the dermis, CD8 in the epidermis. And now for the past several years, we've really been focusing on how these sensory neurons can affect immune cells in the skin. And so the fundamental questions that we're seeking to ask are, what are the effector functions of sensory neurons in the skin? And why do these interactions even exist? And are there ways to manipulate this pathway? So just to set the stage, there are lots of different types of neurons in the skin. And in the lab, we have teams of people working on each one of these 4 groups. But today, I just want to talk about these neurons right here. These are called the very memorable MRGPRD neurons. That's a receptor on the surface, or we can just call them NP1 or non-peptidergic 1. These neurons terminate exclusively in the epidermis. You do find some in the colon, and they're thought to be polymodal, but predominantly sense pressure, like pain from pressure or pressure sensation, not light touch, but like heavy pressure. So our story begins here. This is a really old piece of data. These are the original mice that I made that are missing Langerhans cells, and we did a croton oil assay. This is this is an irritant contact dermatitis assay. And what we found is, unlike Adrian and Clare, who found that their Langerhans cell deficient mice had reduced responses, we actually found significantly exaggerated responses. And this was quite a conundrum for quite some time. And it turns out, we discovered this much later, that it's the fact that the Langerhans cells are constitutively ablated in these mice, which the long-term ablation of Langerhans cells that leads to this increased inflammation in the skin. And if you do an inducible ablation and wait 30 days, you also get this increased inflammation. And so it was kind of difficult to figure out why that might be. But the first indication was we were just looking at the expression of these MRGPRD neurons in the epidermis. This is an epidermal sheet looking down on the epidermis, and red are the neurons. And you can see there's lots of neuron endings scattered all throughout the keratinocytes. The green are Langerhans cells. And then if we spread this to a mouse that has the constitutive absence of Langerhans cells, Langerhans cells are gone, which is good. But you can see the density of these NP1 MRGPRD neurons is reduced. And, we can formally count that and see that it's down. And then just to see if that was indeed the reason why, we generated these mice. These are a diphtheria toxin mediated ablation of this neuron subset. And we did the same croton oil assay right here. And you can see it largely recapitulates the absence of the Langerhans cells. But if we delete another neuron subtype in the epidermis, it doesn't happen. So the croton oil assay is really about proteinuria activating mast cells. I'm just skipping all that data. And if we can just activate mast cells directly without the proteinuria by injecting this mast cell secretagogue called compound 4880, you can see in the mice lacking the NP1 neurons, their response is exaggerated, indicating that mast cells are hyperactive. Interestingly, if you activate mast cells with Fc epsilon cross-linking, you don't see this effect. It's only when you activate them with compound 4880, which activates through the MRGPRB2 or X2 receptor. That's when you see the difference. We, of course, did what everyone does, and we did a transcriptional analysis of the mast cells. And the things to point out here are that the mast cells from the mice missing the neurons have a lot of genes with increased expression. In particular, here's MRGPRB2. This is the receptor for substance P, and it's the receptor that compound 4880 activates, but lots of other genes associated with granule contents and like c-KIT, the growth receptor, are all increased. So these mast cells are hyperactive. What's really fun is that you can activate these NP1 neurons by providing a ligand for MRGPRD. And this ligand turns out just to be the amino acid beta-alanine. And so if we take wild-type mice and just treat them with beta-alanine for 2 days and look at the expression of this activating receptor on mast cells, this is the substance P receptor, you can see that its expression goes down appreciably, but obviously not in the mice missing, but it's already up in the mice and it doesn't have any effect if you don't have the receptor. But if you look at mast cell function, just activating mice with compound 4880, you see you get an increase from the baseline, so more mast cells are degranulated. This is looking at edema in the skin. But if you pre-treat with beta-alanine, you can see that it is noticeably reduced. And of course, this doesn't happen in mice which don't have MRGPRD. So this makes sense. So this actually has clinical implications. So this is a very simple experiment where we do a model of allergic contact dermatitis. This is the DNFB model. You can use other haptens. And this is the line, the dashed line right here. This is about the response that we see with just wild-type mice. However, if we treat the mice with beta-alanine, you can see that we reduce the allergic contact dermatitis response, the CHS response in these mice. And this is mast cell mediated. And you can do the very same thing with another model. This is a rosacea model. This is where you inject cathelicidin. This is Rich Gallo's model. This is cramp or cathelicidin or LL37. And the assay is just injecting this cathelicidin into the skin. And I hope you can appreciate, you get little red areas here. And this is really a model of the flushing component of rosacea. But if you pre-treat with beta-alanine, you can see that this flushing is reduced. And this is quantified right here. So we can agonize these neurons and suppress skin inflammation in mice, and this is mediated through activating these epidermal neurons. And in a lot of work that I don't have time to show, it turns out that the neurons are releasing the neurotransmitter glutamate, and there happens to be a specific receptor on the surface of the mast cell that responds to it. So does this happen in humans? So here's a really simple experiment where we took skin, excess skin from patients undergoing abdominoplasties, and we just took little punch biopsies and we put them in a dish, and we either incubated them with this small molecule. This is SIM2081. This is 4-methylglutamate. This is an agonist for the specific glutamate receptor that you see on mast cells. And then we just came back and added some compound 4880 to the media, and then we just counted the number of degranulated mast cells. You can see here, degranulated mast cell, not degranulated mast cell, and if you count them all, this is the kind of data that you get. So by agonizing the glutamate on the mast cell, which is mimicking the effect of activating these neurons, you see the mast cells are less active, suggesting that this might be active in humans. And then finally what we did is I actually generated a cream. And this is just beta-alanine cream. It's available over the counter. Just mixed it with a bland lotion. And here's a volunteer who did split face application of beta-alanine. Beta-alanine's on this side, and this was the vehicle control. And she has flushing rosacea. And this is a selfie that she took after drinking some wine. And I hope you can appreciate that she has some prominent flushing here on her left cheek. This is the vehicle-treated side. However, in the beta-alanine treated side, you can see the flushing is notably less prominent. And so this leads us really to the final summary model here that I wanted to talk about. So there's two sides when thinking about neurogenic inflammation and neurons driving inflammation. There are these pain-sensing neurons that there's been a lot of work done on, and we've done some work, but a lot of other labs have done fabulous work, really showing that a lot of stimuli in the skin activate these sensory neurons, leading to the release of substance P. And in particular, there's some beautiful work by Nico Denzo showing that house dust mite and other stimuli really require activation of these pain-sensing neurons to release substance P, and that the activation of mast cells is really a critical step in a lot of inflammatory processes. And I've just listed urticaria, rosacea flushing, and dermatitis here, but I think this list is probably quite longer. What I've talked about today is there's this other side of the equation, this other pathway, these MRGPRD or NP1 neurons. These neurons tonically release glutamate, which is the red circle here, that counteracts mast cell function and suppresses the function, keeping it in sort of a homeostatic level. If you delete the neurons, this glutamate is missing and the mast cells become hyperactive. However, if you provide an agonist for this receptor, this is the SIM2081, you can suppress mast cell function, reducing effects in all of these models right here, or you can apply beta-alanine to the epidermis, which activates these neurons to then release more glutamate, which has the same effect. So I guess the take-home point from all of this is that by really focusing on the basic immunology of how these neurons control inflammation in the skin, we have identified a number of potentially targetable pathways that we hope to exploit in the future. And with that, I just want to thank all the people who did the work. There were so many people, no time to mention them all, but I look forward to any questions that people might have. Thank you.
Thank you, Dan, for this wonderful overview of your recent studies. Do we have questions in the chat? Not that I see them right now, or maybe
May I
they're
ask a
coming
question?
in. Dan, please go ahead.
I have a question for Claire. Beautiful talk. Can you tell me how you think the role of TGF-beta plays in forming the identity of these repair Langerhans cells or replacement Langerhans cells in the skin?
So are you asking when we think it's involved?
Do you think it's important, and when do you think it's important, and how do you think it integrates with this jagged signal that you elucidated?
Yeah. So I think, I mean, my assumption is that it has to be important. I mean, obviously from your work, we know that cells cannot be Langerhans cells if they haven't, at some level, if they haven't seen TGF-beta. I think it's a really interesting question. Whether the notch signaling and the jagged signaling is switching on a program that allows the cells to be responsive to TGF-beta, or whether the TGF-beta is then activating the notch, we don't know. The only thing I can say is that we do see in the very early monocytes that come in, we do see expression of TGF-beta receptors. So we do know that as soon as they're in the epidermis, they are responsive. But we haven't formally shown at which point it's required.
Interesting, because what I find so interesting is that these cells are found really at the top of the hair follicle right there, and that's exactly where integrin beta-8 is expressed in the keratinocyte. So I wonder if maybe there's an interaction or a relationship between those two observations.
I think almost certainly. And I think that really precise sort of when we're turning off the more macrophage programs, and then we're activating what we think of as the sort of conventional Langerhans cell genes, and how at precisely what order that occurs, we don't know. We don't actually know what the signal is that switches on EpCAM either, which is I think is a big unanswered question.
Thank you.
There's a question now from Michael Hertwig in the chat. You were studying, Claire, the monocytes that are becoming Langerhans cells, and he's asking whether the Ly6C high or Ly6C low, the non-classical monocytes, are the cells that give rise to Langerhans cells in the skin.
Oh, these are definitely the Ly6C high. So we know that they're CCR2 dependent, and we do capture, I mean, we don't see a lot, but we can still capture some the Ly6C high monocytes in the epidermis before they start to differentiate. Yeah.
There's a question from Mona Homayuni, and it's directed to Dan. She's interested about in your beta-alanine cream and whether it's only used in your study or also used by other clinicians.
Great question. So the person that I showed you before, I made the beta-alanine in my kitchen with a scale, a spoon, and just a lotion that we had in the cabinet. Since then, we've gotten more serious, and it's actually being licensed by a cosmetic or consumer products company. And so they're in the process of developing it. So I am hopeful that at some point it will be easily available commercially. But at the moment, it's just, you have to make it yourself.
Okay, great.
I would, or maybe I go ahead. I would have a question to Clare. We are now talking a lot about immune memory of the skin. Do you think the change in the Langerhans cells, the old Langerhans cells or embryonic Langerhans cells versus now then the monocytic immigrants make a change in the immune response in the skin so that the skin remembers this insult?
Yeah, I think this is a really interesting question. I have to say that when we started the work, all my money would have been on the fact that, yes, that there is a difference in these cells because they're coming from adult bone marrow cells and they're in a different environment. But at the moment, we haven't been able to find a difference in these cells. That said, I think without question, in patients who've had GVHD and recovered from GVHD, we know that they have sort of lifelong problems frequently in the skin. And I think without question, there is a prolonged disruption to the skin barrier as a result of the pathology. And whether the Langerhans cells are contributing to that, we don't know. But I think almost certainly that's the case. Yeah.
Can I follow up on this? So let's say someone has an allogeneic stem cell transplantation, and that might create a niche because of cell loss in the skin, and then you have your monocytes becoming this new immigrants. Is it possible to still identify a host Langerhans cells from the time before transplantation simply because they persist chemotherapy, and then detect the old ones with a macrophage-like profile and compare them to the new ones based on single-cell chimerism, and then compare them within the same site, maybe by single-cell transcriptomics to find out whether there are differences in their regulatory identity?
So I think it's becoming possible. So I think simplistically, I mean, people like Matt Collin have used the approach of using fish. So when they have sex mismatch transplants, they can track Langerhans cells that are coming from the donor or the host based on the presence of male antigen. But that's just at the level of fish. We haven't been able to do that till now because our new Langerhans cells, as far as we can tell, look transcriptionally identical to the cells they're replacing. There's no evidence of the different origin. But I do think that we're probably now at the point with the new advances in especially like single cell, like sequencing, and that we will be able to actually try and answer some of those questions. I think the question of whether embryo-derived Langerhans cells persist in old adult, as we age, and to what extent monocyte-derived cells come in is a really important question. One thing I would add is that the equivalent macrophage population are the microglia in the brain. So it was always thought that they persisted indefinitely as embryo-derived cells. And now increasingly, we're finding that that may not be the case, that very late in life, they do slowly get replaced by monocyte-derived cells. And my money would be on the fact, I don't know what Dan thinks, that this probably occurs in human skin as well, but we just don't have the tools to detect that yet.
Maybe I would have another question for Dan. I think in the beginning you showed that these dendritic cells, that the Langerhans cells are also important for the sensory neurons, to have them there. So do you think by means of, I think, UV irradiation, you reduce Langerhans cells? Do you also reduce then the sensory neurons and thereby maybe get some effects of the therapeutic response due to UVB radiation? Do you think there's a kind of circuit?
Yeah, I think it's a very interesting idea. I think the fact that these epidermal neurons are dependent on Langerhans cells highlights their macrophage function. And the way it works is they're probably secreting a growth factor that's required for the neurons to terminate in the epidermis. So UV light could certainly be used to ablate the Langerhans cells, but they would need to be gone for quite some time for the neurons to be removed. In our hands, in mice, it takes about a month following complete genetic ablation of the neurons. I would worry that UVB does indeed reduce the number of Langerhans cells, but as Clare and others have shown, there's a compensatory recruitment of these new Langerhans cells that come in fairly quickly. So I don't think you'd get into a state where there is a sufficient absence of this Langerhans cell monocyte-derived neuron growth factor to lead to a resolution in the sensory neurons. And I'm not entirely sure why you would want to get rid of these sensory neurons, right? It's not, there are plenty of other neurons that can sense pain and itch. So I don't think you're going to have clinical benefit by doing that.
Okay.
Can I ask on that, Dan, so in your mice that never have Langerhans cells, so presumably they never have properly innervated epidermis.
Yes.
Have you ever looked at the physiological consequence of that in terms of sensation?
Yeah, we should have. The reviewers oddly never asked. So no, we haven't done it with the Langerhans cells. Sort of, we made the observation from the Langerhans cell mice. That's what got us started. And then it was too complicated, so we just moved to the neuron ablation mice. And that's where all the studies were done. But we haven't circled back to do the Langerhans cells. And it's an interesting point. We probably should have done that.
And maybe along the line, has anyone checked what would happen in settings of Botox injections that are done on a regular basis?
Yeah.
Are not all neurons somehow paralyzed, but you have those substance P versus, the glutamate, secreting ones?
Fantastic point. So Botox, which all dermatologists, love, it prevents the fusion of vesicles at the neuron terminal, so neurotransmitters are not released. So if you're treating wrinkles, you inject them into the facial muscle, so it's a relatively deep injection, right? So in those injections, you paralyze the motor neuron junction, but the Botox isn't up in the skin, so it doesn't have so much of an effect. If you inject Botox into the skin, which has been done sort of in studies, you actually can improve psoriasis and atopic dermatitis, because these diseases require continual neuronal input. And then when the Botox wears off, the disease returns. And there are also lots of examples of patients who have had nerve damage from a surgery or a trauma. And there are anecdotes where their psoriasis and atopic dermatitis resolves downstream of this loss of sensation, but returns when sensation is restored. So I think that's pretty good evidence that these sensory neurons are important in maintaining cutaneous inflammatory diseases. And I think the challenge that we have is sort of sorting out the different roles of individual neurons, right? Different subtypes of neurons, because there's some that make substance P, there's some that make CGRP. I've talked about glutamate, but there are more things on the menu. There are more neuron subtypes and there are more neurotransmitters, and sort of understanding how they work together on different cell types in concert. I think that's the challenge.
Yeah. And do you think they might even be causative for these diseases? If they get better when you paralyze the neurons, could maybe a nerve abnormality or a particular phenotype or SNP even be a cause for psoriasis in these diseases? Or do you think it's just exaggerating pre-existing skin disease? Because neurologists would be excited if this is now becoming a neurological disease.
It seems to be important in maintaining the disease for sure, and possibly for triggering it as well. But if you if you think about the SNPs associated with psoriasis, they tend not to be neuron type things, right? So I wouldn't say in the case of neuron or in the case of psoriasis or eczema that the primary genetic defect lies in genes expressed in the sensory afferents. I think that's unlikely, but I think it reveals that these neurons are part of an inflammatory feedback loop. And what that provides is like a whole new set of targetable pathways that will not be immunosuppressive.
Yeah.
And I think that's the exciting promise of this.
Yeah. I think we have one final question. Knut, do you want to read it?
No, where is it? No, you mean
Or I'll just do it because
alanine we had already, and then
Ian Odell. Mast cells are increased in scleroderma skin. Any role for NP1 mechanosensation in driving fibrosis? Question for Dan.
Fabulous question. I don't know the answer to it.
Okay, so this needs to be explored.
Yeah, I mean, we're actively working on like exactly how this works, how the factors are related. And yes, the interaction with fibroblasts or myofibroblasts, super interesting.
Fine. So I think it's time to close our session here today, Sweet and Sour. I think with Claire and Dan, it was a very fruitful, wonderful discussion. Also having you two with the two sides looking at Langerhans cells and their function, regulating different aspects of the immune response in the skin was particularly intriguing. Now, thank you very much to you two sharing your insights here today. And I also thank my co-chair Christina for having this meeting here today. And there's more of an announcement today. And the ESDR Kitchen Freshly Baked will start the next one on the 1st of July with Lauren Juin-Massé and Paula Restrepo. And there is more. The ESDR Kitchen webcast is then open access on YouTube, so you can see it again there or share it with friends. And of course, we will have the ESDR here in Heidelberg this year in the beginning of September, and you can still submit abstracts until June 24th, June until 12th of July.